All right, guys, next up today is what are we actually using DNA for? So transcription and translation, aka the central dogma, aka protein synthesis. You're going to need to be able to recreate this chart. So you could be asked to compare DNA and RNA. Note you need apples to apples. So on your last test, some of you guys missed points because when you were comparing anaerobic and aerobic, you didn't have an equal statement for both. So if I say shape, I need a statement for RNA and a statement for DNA. So when I talk about comparing DNA and RNA, when I say shape, DNA is a double helix, it's 2 strands. RNA is single-stranded. When I say sugar, DNA is deoxyribose, RNA is ribose. Bases, DNA's got adenine, thymine, cytosine, and guanine, whereas RNA's got adenine, uracil, cytosine, and guanine. Apples to apples comparisons, just make the table. We have 3 types of RNA. 2 you learned about freshman year. So there's transfer RNA, aka tRNA. It's folded upon itself and it carries the amino acids to the mRNA. And then messenger RNA, the mRNA, aka mRNA, sequence of nucleotides that determines the primary sequence of a polypeptide. Then there's this third kind, this ribosomal RNA, or rRNA. This makes up your ribosome. So here is a picture of your ribosome. When you look at it, this is the location of translation. It is composed of rRNA and a protein. It's got a large subunit on top and a small subunit on the bottom. That small subunit is where the mRNA is going to kind of land. It's the holding place for it. The mRNA and the tRNA meet up in the space between the 2 subunits and will often group together on a single mRNA to speed up the process. So when you guys learn it freshman year, you think like this one little Pac-Man of ribosome is moving down the mRNA doing translation. Instead, you could have hundreds of little Pac-Men moving down it. And so you're producing these proteins very, very quickly. On the mRNA, site A, which is this first site right here, holds the tRNA with the next amino acid for the polypeptide chain. Site P, which is the next site over, holds the tRNA with the last amino acid that was added to the chain. And then site E releases the tRNA that has lost the amino acid. 2 different types of ribosomes, free and bound. Free ribosomes are making proteins and enzymes that will be used inside the cell. Bound ribosomes are making proteins and enzymes that will be used outside the cell. The bound ribosomes are bound on endoplasmic reticulum, and they then have the polypeptides enter the lumen of the rough endoplasmic reticulum that then gets transferred to the Golgi apparatus, which gets packaged into a vesicle for export out of the cell through exocytosis. First step, transcription. If you're going to transcribe something, you're going to rewrite it. And note that this is tightly controlled. Remember, your cell, your DNA, contains all of the instructions to make everything in your body, but each individual cell only needs to access a small portion of that. And so we are going to control transcription with the use of our histones. The more tightly wound stuff will never get transcribed. Methylation prevents the transcription of maternal or paternal forms of that gene. Remember, you've got the mom gene and the dad gene. We can hide away mom's gene and only transcribe dad's gene. Pattern can be linked to cancer for early detection and personalized cures. And this is that whole idea of epigenetics. So which genes are used and which genes are not used. Make sure you watch that video on epigenetics. Sense and antisense. Only one strand of the RNA is transcribed. The sense strand is the strand that is not transcribed. The antisense is. The mRNA that is produced is complementary to the antisense strand of DNA and therefore almost identical to the sense strand. If you think about it, you're going to send this message out. So here is the message that you want. Here is the opposite of the message. You're going to transcribe the opposite of the message so the mRNA pretty much mirrors the message. And if I can figure out how to do it at the end of this lecture, I'll draw it all out, and that might help. Or I might do that in person. Why don't we do that in person? So when we meet on Friday, I'll draw this for you. Transcription in eukaryotes. The first step, RNA polymerase binds to the DNA at the promoter region. This will start the transcription process. The promoter region is usually a Tata box. It's a repeating sequence of nucleotides that are like this big neon sign that says, hey, the gene you want to transcribe is over here. The second step is the RNA polymerase links the free nucleotides working in the 5' to 3' direction, present in the form of nucleoside triphosphates. We've seen that before. Temporarily bond to the antisense strand, acting as a template. As the RNA is formed, it separates from the DNA, and the hydrogen bonds between the sense and the antisense strand reform. This process continues until a base sequence called the terminator is reached. Here it is in pictorial form. So we have the, hey, transcribe here. And then we have it break apart. You're transcribing the antisense strand, and then you end up with your mRNA. At this point, you have a piece of mRNA and it needs to be modified. And so we're going to remove those introns, those pieces of information that don't have what we need. So the mRNA after transcription contains both introns and exons. Post-transcription modification removes the introns, and the enzymes catalyzed cut out the introns and glue the exons back together. We call this process splicing. So removing the introns and gluing the exons back together is splicing our mRNA. This new strand of mRNA is considered mature, and we are going to put a cap and a tail on it. A cap on the 5' end and a tail on the 3'. This protects it from all of the enzymes that are in the cytoplasm. So as those enzymes want to eat away at this mRNA, it's got a buffer zone between the stuff and the message. Translation is the next stage. So we're going to translate this mRNA into a protein. So what happens at the ribosome, composed of a large and small subunit made of rRNA proteins. 3 sites, like I mentioned before. The A holds the tRNA with the next amino acid. The P is the growing polypeptide chain, and the E is the ejection seat. So it ejects the empty tRNA. Codons are based on sets of 3 nucleotides read from the mRNA. 64 different possible combinations. There are only 20 amino acids, which means you could have multiple codons code for the same amino acid. This helps prevent mutations. tRNA amino acids are activated in the cytoplasm. At this point, it becomes tRNA and it combines at the 3' end. Requires energy in the form of ATP to form and attach to the tRNA. The amino acids are attached by enzymes, and each amino acid only goes on a specific rRNA. Helps ensure that the correct amino acid is used in building the polypeptide. Here's what it looks like. So at the bottom on the loop here, you're going to have your anticodon that is going to be complementary to a specific codon. On the opposite end, attached to the 3' is going to be your amino acid. The first step, the tRNA is activated. The second step, the mRNA binds with the small subunit of the ribosome. We call this initiation. We are initiating the process of making your protein. Happens when the ribosome encounters AUG. AUG, or methionine, is always the first codon that allows for the building. Third step, translation occurs with the help of the large subunit at the 3 different sites. A for the incoming tRNA, and then P to attach that tRNA's amino acid to the polypeptide chain, called elongation. We're elongating the polypeptide. And then it ends at E, where we eject the empty tRNA, or termination point. Pictorial form again, take a second to look at this picture. So the initiation of translation. The ribosome attaches to the mRNA. The tRNA must be activated using energy to attach an amino acid to the tRNA with the help of an enzyme. The rRNA of the small subunit will recognize and attach to the ribosomal binding site on the mRNA near the 5' end. The large subunit containing the tRNA binding site attaches, and the first tRNA binds at the P site and begins the process. Then it's going to start elongating our amino acid. When the first tRNA binds to the ribosome, it changes shape, opening up the A site for the next tRNA to bind. The first tRNA codes for methionine and is considering the start codon. Memorize AUG as the start codon because they're not going to tell you that on the chart. It will just say AUG or methionine. You need to know that's the start codon. The second tRNA will bind at the A site. The first tRNA will be released and the first amino acid will be linked to the second by a polypeptide bond. The ribosome moves 3 nucleotides towards the 3' end. The first tRNA minus its amino acid is now located on the E site on the ribosome. The second tRNA is now located at the P site, and the third incoming ribosome is located at the A site. We call this translocation. Finally, we get to termination. In termination, it's going to occur when the ribosome moves over one of the 3 stop codons. These stop codons don't code for any amino acid, and so it's an empty spot, and that signals that we are done with the process, that it is over. The polypeptide's released from the ribosome, and the ribosome disassociates into its individual subunits. There we go. Okay. So these are approved IB proteins and functions. These are the ones that you need to know for the IB exam. It could show up in the multiple choice. It could also show up in a short answer where you have to write about an example. So hemoglobin contains iron and transports oxygen and carbon dioxide. Actin and myosin are in your muscles. Insulin is regulating your blood sugar. And the amylase is found in your spit to break down starch into simple sugars. Immunoglobulins are in your immune system. Here are the videos that you need to watch. They're going to tell you all about this process.